So our next presenting company is Semma Therapeutics, and it is a pleasure to introduce Rob Millman, who I first met, I think, at Celera, believe it or not, all the way back when. And then later we worked together at Alnylam. So it's really a pleasure to see you and to reconnect. And it's been exciting to kind of learn about Semma a little bit in just diligence in the company in preparation for our conversation. So I think Rob has a few slides that he's going to run through, and then I'll ask you a couple questions and certainly open it up to the floor.
Great. I don't need to tell you that diabetes is a worldwide health epidemic. In the U.S. alone, the number of insulin-dependent diabetics is over 6 million people, and it represents over $700 billion in the healthcare burden. In a typical patient's life, it's not that easy. Insulin, which was discovered and administered first in 1921, is coming up on its 100-year anniversary. Typical person has to prick their fingers 4 or 5 times a day, insulin injections. This is what a young woman would have to deal with. And even with insulin therapy, A1C, which is a measure of the, how well you're controlling your blood glucose, you can only get down to about a 7 at this level, and you're still getting nerve damage, limb damage, eye damage at that level. Can you cure diabetes? And the answer is yes. It's not long-term durable, but it's been shown to be curable. How? Pancreatic transplants, and then more recently in the late 90s, islet transplants. Graph A is what a person's blood glucose looks like under control using glucometer as well as insulin injection. But when they receive islets from a dead person, which then the person is also on immunosuppression, so they're not rejected as a foreign tissue, glucose levels are maintained. How long does this work? Well, the recent trial results were published from the US trials, and these went out 5 years for 50% of the patients. One study site said 7 years, and this is an islet preparation that's isolated from a person who's died. The organs isolated, the cells are isolated from that, and then they're shipped. And so these are really treated harshly. And even with that harsh treatment and in the presence of immunosuppression, which kills most beta cells over long terms, we've got long-term survival. We've met patients who've had islet grafts 20 and 30 years ago, 20 and 25 years ago, and are insulin independent still. So the prospect of starting with a pristine islet preparation, one that's almost juvenile in characterization and able to grow with the patient, would be very encouraging. And that's really where we came from. Is the idea was very early, a cartoon. Can you take a stem cell and differentiate it to the islet, just like a cadaveric islet, and put that in some sort of protective mechanism and put that in the patient? If you were able to do that successfully, you'd be able to obtain glycemic control. And that was really the focus of our founder, Dr. Melton, whose 2 children, Sam and Emma, were diagnosed with diabetes almost 20 years ago. He decided to focus on this final pathway to reach the final differentiated beta cell, and published in 2014 a seminal article to produce functional beta cells. And we've been able to move that technology in-house and scale it. Sam and Emma are still very active young adults, and Sam and Emma became the motivation for Semma Therapeutics.
Great, excellent. Well, thank you for that overview, and I'm going to ask a couple questions, but this is a pretty exciting story, so if there's any questions from the audience, don't hesitate to wave and raise your hand. So I guess I want to start, and I think I know the answer, but I'm going to ask it anyway. What really is the secret sauce in making these islet cells? I'm sure there's a lot of learning that takes place to make these things viable, but what can you tell us about your process?
Well, it was a very nice publication in the Cell article in October from Dr. Melton that outlines the process. And there's been also a couple of papers that have appeared at the same time and subsequent for other ways. Basically, you're trying to mimic what happens in 9 months of embryogenesis from a single embryo cell leading to all the cell types. You're trying to mimic those signals to get a cell to go through that process to get to the final cell process. Let me go back. And so really the goal was to be able to take a stem cell, move it through differentiation to the final process. And the field moved very quickly through to about 2005 and got to the point of a pancreatic progenitor. Dr. Melton then did combinatorial analysis of the various pathways that are integrated through the embryogenesis of a beta cell and looked at what factors are needed, what factors are not needed, and came up with this universal cocktail of basically 6 steps to lead to a 30-day differentiation process to make the beta cell.
Excellent. So that was more detailed than I was expecting. So I appreciate that. That's helpful. You mentioned that ultimately these islet cells will be contained or encapsulated in a device to enable vascularization and insulin secretion while protecting those cells from the host immune system. How do you envision this working, and has Semma selected a go-forward construct at this point?
Yeah, the simplest way we could think about replacing the cadaveric islets with ours would be to use them in the same situation as a naked cell transplant with immunosuppression. Lifelong immunosuppression is something that we'd like to avoid, but that is a path forward. However, what we decided to do is approach this by providing an immunoprotective barrier to allow glucose to come into the cells, oxygen to come into the environment, insulin to go out, but not the bad things that kill the beta cells to come in. So we've set out in a process and a program to develop these devices that will be based on perm-selective membranes that will allow the ability of the cells to thrive in an environment in the body, yet be able to function like a beta cell and not be rejected as foreign tissue. It's actually a very complex process that starts from membrane manufacturing, whether it be electrospinning or stretching of ePTFE, membrane coating to make these hydrophilic, which are normally hydrophobic, putting in specialized device housing so that they don't have sharp edges to cause microtrauma, finding the right surgical place. We have robotic assemblies that are done in GMP suites, basically bioisolators, and finding a way of filling and finishing these products to make them ready is a complex engineering process. But we had the fortune of teaming up with a group, purchasing a group called Cytosolve in Providence, Rhode Island. It was a group that developed delivery technology for a company called Neurotech. They've had devices in the eye for over 10 years without any fibrotic events. They developed alginate microspheres for living cell technology, which was bought by Otsuka, and that had porcine islets, didn't establish diabetic control, but showed survivability of the islets. They've developed devices that sit in the brain as well as a spinal channel. So this was a perfect project for them to work with. They started with the cells in the environment and developed a device de novo from scratch with us. And we're now in the process of selecting the final membranes as well as the device designs to allow perfect vascular access to our cell components.
Great. And Rob, what does the path to the clinic look like and ultimately approval? And I guess we've been talking a lot about the 21st Century's Cure Act today, so maybe we can kind of weave that into your answer, whether that shortens the way.
Yeah, this is going to be regulated by CBER as a drug, but with the Advanced Cure Act, the FDA will form a new group. The device group comes in and sits on the advisory team. We've had our first meeting with the FDA on a pre meeting to establish what our clinical path looks like, our cell line selection, our device containment. They're very happy that a device is used. Longer preclinical tox studies are needed if you're going to naked cells, as was experienced by Asterias. The proof of concept has been established with cadaveric islets. We're looking for glycemic control or time within a proper range. The number of patients are very few. The average trial size is 10 to 15 on the phase 1, phase 2 second site expansion to a similar number, and then a registration trial has been suggested in cell therapy to only need about 40-45 patients. The readouts will be very quick. Our cells will come into the environment very active. An example being is animal data that basically within 2 weeks after establishing a graft, there's a little device in a fat pad, these cells are actually able to engraft and start to function quite well. In this particular experimental graft, basically we induced diabetes with a chemoinduction agent. We used insulin pellet to control the diabetes. We implant devices, and then when the devices are either removed or in deviceless confines, the animals go diabetic and die. But we're able to establish fairly tight glycemic controls very rapidly with both our cells as well as cryopreserved cell preparations.
Great. And it's interesting, too, because if you envision this, it's not only an insulin pump, but also a reader as well, or monitor as well. So, and also the output is really insulin. It's very easy to measure. So how does that sort of benefit the regulatory process or the clinical development and regulatory process?
Well, I think it makes our clinical pathway fairly straightforward. We're looking for a very quick signal, and if we can hit the home run, actually glycemic control on the first few patients, it'll be a transformative event. Since the signal of what we're looking for, human insulin, in a patient that doesn't normally produce insulin is a very easy readout, we won't need to be doing placebo controls for this. We can use historical data both with that patient and the population as our control arm, and therefore it's a very focused study event. We don't have to do sham surgeries or implantations of devices without cells. This is just looking for efficacy signals.
And I do want to ask, we've had another company present today called ViaCyte, which is working in a similar space. How is what you're doing different? And I know that you've spent a lot of your career focused on intellectual property, and ViaCyte spoke about the Beta-Logics merger. So how does the IP shake out in the space?
When somebody gets a product to the market, it's going to be a big shakeout. I think that, ViaCyte spent a long time and was very successful at moving forward a program based on this earlier cell type, the pancreatic progenitor. We've hoped to see, and we hope to continue to see clinical successes from them because it'll only help both them as well as the industry as a whole in bringing attention to the fact that cell therapy isn't something that will always fail, but will actually succeed sometime. IP, it's a public forum. We can't really talk about IP, blocking IP, but again, our work is focused around two focused areas. One is the intellectual property developed by Dr. Melton and the terminal steps of making the functional beta cells, as well as the device work. And I think that in the device work, we've been able to come up with brand new chemistries involved in membrane forms that will have efficacy in other industries, even in filter industries. But we'll use this as a basis platform for any hormonal cell production type therapy that can be used with a device or a device strategy.
Yeah, I was going to ask because it really, especially with this expertise from the company that you acquired, you really could start to look at other cell, protected cell populations for other diseases. So just sort of kind of looking out to the future, what else makes sense to you?
Well, I think for us, we're focused on diabetes, and to tell Doug that we're going to cure parathyroid hormone refractory syndrome as opposed to diabetes, his children and him would say, what are we doing this company for? So we're focused on diabetes and see really that Semma itself, the next generation, will be looking at modified cells, potentially making the universal cell approach or knocking in checkpoints, PD-L1, LAG3, CTLA4 to avoid immune surveillance. We had a California Institute of Regenerative Medicine grant, as well as a local, what we call the BERT program, looking at autologous. I'm a believer in autologous therapy, but unfortunately the cost of a GMP iPS cell from a person is still cost prohibitive. But as systems are developed for closed loop productions in the $5,000 to $10,000 per range, you could think of a personalized medicine approach. You can think of next generation cell approaches. As for other cell environments, again, the low hanging fruit, parathyroid hormone, any cell factory, or if you have a cell that needs to sense and secrete, this is a type of device that would work well
very
for
helpful.
that.
Question from the audience.
Thank you. I just said congratulations, Ben. And can you comment on whether the agency thinks that you'll need to demonstrate that you can retrieve the device and that a post-retrievable state that, in fact, the cells are still doing only what it is that you want them to do and not something else?
The agency doesn't put requirements on you. You come to the agency with your plan and they tell you whether it's sufficient or not, or just tell you very few things and then you hope that it was sufficient. And so we went forward with a encapsulation retrievable device, which we thought provides a safety window in case the cells do turn into cyst ducts or if something worse, you'd be able to pull it out, it would be contained. So we'll need to show both biocompatibility of the materials plus that the cells can't escape from it, and that'll give them the safety signal that will give them comfort. Beyond that, the function of the cells after you retrieve the device, they're not worried about that. What they're more concerned about is how long will this work? And again, that's something that we'll design as a trial basis, as a 1-year follow-up study, but then a follow-up of the patients to see how long they go. We just don't know. However, we do know that hernia meshes and other biocompatible materials are in human bodies for 20, 40, 50 years without huge fibrotic insults or events. And so by using that first principle of how do you make a material stealth or acceptable by the body, and then use that as the material basis for your device, I think gives us the ability to think that this will be a long-term durable solution.
Great, excellent. Rob, thank you so much for being with us today. Really cool stuff. I look forward to hearing more.
Thank you very much.